Hello again, biology students. We're ready to continue our talk on genetic engineering. Today I want to talk a little bit about a concept called recombinant DNA. If you look at the word recombinant, you can see that it has the word recombine. So we're basically taking DNA from one organism and we're combining it together with the DNA of another organism, and we're looking for the protein products that result from that. And so what we'll be looking at today: how do scientists copy the DNA of living organisms? How is recombinant DNA used in the real world? And how can genes from one organism be inserted into another organism? We do have some vocab over here. We will be talking about what each of these are during today's talk. I also wanted to remind you that there is no homework due for this section, so don't feel like you have to do it. So let us go ahead and get started. I want to start with a funny little cartoon. We have a female and a male scientist, and so she's looking on and he's explaining what he's doing in the lab. He's saying scientists on Earth are attempting to genetically engineer a cat with non-allergic fur. And so for those people who are allergic to cats or dogs, one of the ways that genetic engineering can be used is to figure out a way to make cats that don't have any non-allergic fur. And so he, however, wants to go one step further. He has decided that he will genetically engineer a dog that will not drink from the toilet, because that's just gross, right? And so she says, well, then why don't you just put the lid down? And so he says, putting the lid down is not advanced science. And so we have to be careful, because just because we have the technology, the science in order to do something, it doesn't necessarily mean that it's the right thing to do. And so we would refer to these types of problems as bioethics. And so we'll talk about that in just a second. Until very recently, and I'm going to say within the past 40 years, breeders couldn't introduce new changes to living things. The changes produced in the DNA were random and unpredictable. And so basically, you would mate different plants, you would try to get different offspring, and hopefully if you got an offspring with a desirable trait, then you would try to mate that to another plant or another animal and try to create more of that. But it was very sporadic and it was very random, so there was no controlled way to do that. However, because we now have information regarding the genetic code, so remember, the genetic code is what allows scientists to translate the three-letter codons of the messenger RNA into the individual amino acids that make up a larger protein. We can use that information to know what type of protein we're dealing with. Scientists can modify DNA at will, and so what they typically do, they'll take a gene of interest, which again, as you remember, genes are made up of DNA. They'll cut out the DNA, they'll then identify the base sequences, and then they need to make lots and lots of copies of the DNA. And so we'll talk about what PCR is here today as well. As we looked at the cartoon, even though this guy wants to make a genetically engineered dog that doesn't drink from the toilet bowl, she brings up the good point: why not just put the lid down to prevent the dog from doing that? And so, again, we have to always ask ourselves, just because we can do something using science and technology, is it the right thing to do? And so, bioethics is the study of typically controversial ethical issues emerging from new situations and possibilities brought on by the advances of biology and medicine. So again, I want to point out that science in of itself is neither good nor evil. It just depends upon how people use the technology. And so that is really then what's at the heart of bioethics. And so as you can imagine, things like human cloning is a very, very big bioethical issue. And so we'll talk a little bit about that also later on. We're going to go ahead and define what genetic engineering is. It's the process of making changes in the DNA code of living organisms. And so typically when we genetically engineer an organism, we're trying to make or create a protein that will be somehow beneficial to humans. And so, for example, diabetics oftentimes have to inject themselves with insulin. The insulin that used to be gotten from cows' pancreases is now gotten through bacteria. And so the genetic engineers are able to copy the gene that's responsible for making the insulin, put it inside of a bacteria, grow up the bacteria in multiple, lots and lots of numbers, and therefore those bacteria will then produce the insulin. And so those pens that you see diabetics use is the type of genetic engineering that does serve a practical application. All right. I want to talk a little bit about the tools of molecular biology. Unfortunately, we will not have time to do any of these types of experiments. If you decide to take AP biology in the future, a couple of the labs will involve the use of gel electrophoresis. And so we'll be looking at some tools right now of molecular biology. Before you do any sort of genetic engineering, you first have to extract the DNA of interest. And so what you typically do, you grow up a culture of cells, and then you basically break them apart. You can use sound, you can use cold. There's other techniques you can use to do this, centrifugation. Then the DNA is separated from the other cell parts using centrifugation. And so if you think about the dryer in your laundry room, the dryer is spinning around very, very, very fast in order to drive the water off of your wet clothes. Using a centrifuge, scientists can spin down a sample of cells. The DNA will come out at a certain part based upon the speed of the centrifuge. Scientists then can extract the DNA from that and then get rid of the cell waste from that point. All right. Then, in order to get to the specific DNA sequence of interest, we use a specific type of enzyme. And so remember that enzymes are catalysts that are protein in base. And so these enzymes will cut the DNA at specific base regions. And so you not only do it to the donor cell, but you do it to the acceptor cell as well. So you're basically able to cut and then paste in the gene or DNA of interest into a particular organism. Then, what you oftentimes need to do, you need to identify what those products are that you've cut, because not all the products that are going to be cut by the restriction enzymes are useful. We use in the lab a technique known as gel electrophoresis. What you basically do, you take a mixture of DNA, you place it at one end of a porous gel. So porous gel means that there's tiny little holes for the DNA to travel through. And so we apply a voltage down here. And so notice that the fragments are going to go from the negative end to the positive end. The reason for that, if you remember the structure of DNA, if we look at the backbone of DNA, remember it's made up of sugars and phosphate groups. And remember, the phosphate groups are negatively charged. The negatively charged phosphate groups in the DNA will naturally be attracted to the positive anode, because remember that opposite charges attract. And so that's why the DNA is going to go from the negative end down to the positive end. And typically what happens, the smaller DNA is going to go through the gel faster, and it'll get further down the gel. The larger pieces of DNA are going to get held up, and so they're going to not be able to separate as much. So if you look over here, you'll see that the shorter molecules are further down on the gel, whereas the larger molecules tend to remain at the top. And so these, again, are some useful tools that molecular biologists use in the lab in order to help with their experimentations. All right. Once we have the DNA, then we can sequence the DNA, and we can then even change it at will. If we know the sequence of the specific DNA, we can compare them with other organisms' genes and discover the functions of different genes. And so we call this process aligning. And so typically, genes that are essential for life, you're going to find that they do not change between different organisms. And so, for example, if you look here, you're going to see a codon that is AAG. And if you look, we have a total of 23 different species. We've lined up the different sequences of DNA. You'll notice that, for example, this sequence here and here, amongst the different species, there is no change. And so whatever proteins that these particular genes code for is essential for all of life, because notice that there are no mutations or relatively few mutations, so most of the bases have not changed into different bases or been inserted or deleted. And so we do see that highly conserved genes are genes that are essential for life. And so over here, you're going to see that these genes maybe are not as essential, and therefore you are going to see between the species that there are some differences between the nucleic bases in certain parts of the DNA. And so by doing this, we can make comparisons with other organisms to then understand the functions of different genes. All right, and so I talked about the fact that you're able to read the sequence of DNA once you break it down into the small parts using the restriction enzymes. What scientists have done, and this is actually very clever, remember that in DNA there are the 4 bases. We have adenine A, cytosine C, guanine G, and thymine T. There are compounds called fluorescent dyes that under UV lights will shine a different colored light. And so what the scientists have done, they have a different fluorescent dye added to each of the 4 different bases we find in DNA, and therefore they complement the template strands. So remember Chargaff's rules, A goes with T and C goes with G. And so we could use this then to shine a light on the fragments after we run them through a gel electrophoresis. And then when we shine a laser beam on it, we can then get the specific order of the bases using the different colored fluorescent dyes. And so a very, very clever way of then deducing what the specific DNA sequence that we're interested in. All right. I mentioned before that you need to, one of the earlier steps in genetic engineering is to be able to cut out a region of interest for DNA and then pasting that into another organism. And so we can see that short DNA sequences can be assembled using a DNA synthesizer machine. And so before it was done by hand, it was done by a particular molecular biologist, it was very labor intensive. They have since developed machines that will do this automatically so you don't have to worry about it. Synthetic DNA is then the combination of natural DNA joined together using DNA splicing enzymes. So to splice is to cut. And so the DNA splicing enzymes will cut out at the specific regions of the DNA of interest, and then you can combine them together. This results in recombinant DNA. We're recombining the DNA from different sources, and so we can then put them together. I want to talk a little bit about this concept of plasmids. If you remember, in prokaryotes, when you look at the DNA is typically one large circular chromosome. And so that is basically what a plasmid is. So this is a plasmid, and so this is from a bacteria of some sort. Notice there's a couple interesting things. We have in this part of the DNA, we have our gene of interest. We also have a gene that causes resistance to a particular type of antibody. And so the first thing we need to do is we need to join the foreign DNA to the small circular DNA molecule, which again we refer to as a plasmid. The plasmid DNA, as I just discussed, has two specific features. We have a DNA sequence that helps to promote plasmid replication, and then this is our genetic marker. And so what this allows us to do is to distinguish bacteria that carry the specific plasmid from bacteria that don't carry the plasmid, the specific gene. So for example, if this was like an insulin gene, I have an antibiotic resistant gene, I grow up the bacteria, I treat the bacteria, in this case with a kanamycin. The bacteria that have this particular genetic marker are not going to die because they have the resistant gene, which has my gene of interest. However, bacteria that do not take up the gene of interest are not going to have this resistant gene, and therefore if I subject them to the antibiotic, they will die. So only the bacteria that have my particular gene of interest will survive the antibiotic treatment. And so this is another form of technology that helps us select the bacteria that produce the particular gene of interest. As I mentioned before, we not only need to cut and paste the DNA together, we also need to make lots and lots of copies so that there's a good chance for them to be able to be incorporated into the plasmids. And so we have a technology known as polymerase chain reaction. And so polymerase in this case is referring to DNA polymerase, which again is the major enzyme involved with DNA replication. So making more DNA, the main enzyme again is DNA polymerase. All right. And so there are 3 steps in this process. The first step is known as denaturing. This is when you heat up the DNA, and that's going to separate out the double helix into the single strands. And so by heating, you're going to separate the 2 strands of the double helix into 2 single strands. Once that's done, you're going to cool down the DNA, which allows the primers to bind to the single-stranded DNA, and then we use the all-important DNA polymerase enzyme to add the complementary strand to then elongate the DNA. Then we go back and repeat the process, and so from 1 double helix, we can get 2 copies. Then from 2, we end up with 4. From 4, we end up with 8. And so there's this doubling effect that happens every time you go through these 3 steps. The PCR machine here, you can program it so that you can let it go overnight. And so when you come back in the morning, you will then have the many, many copies of DNA. And so I want to also take then a moment to talk about the inventor of the polymerase chain reaction, also called PCR, a gentleman, American, by the name of Kary Mullis. And boy, is he quite a character. All right. This gentleman invented polymerase chain reaction in the year 1983, so about 30 years ago, while working for a company in California, the Cetus Corporation. He did, in fact, win the Nobel Prize in Chemistry in 1993 for developing this work, but I kind of wanted to point out a few little quirks about this gentleman. He was camping out in a woods in Northern California and claimed that he saw a glowing green raccoon. Yeah, okay. And so if that doesn't convince you that this guy's maybe a little bit off, he's also the only Nobel laureate that has claimed to have been abducted by aliens. And so yes, this guy has said that aliens did abduct him as well. He also was able to try to turn this into a profitable business. In the year 1992, he founded a business with the intent to sell pieces of jewelry containing the DNA of famous people like Marilyn Monroe and Elvis Presley. and people like that. And so the reason I bring this up, even though this technique nabbed this guy the Nobel Prize, just because you're brilliant doesn't necessarily mean that you're quite all there, if you get my drift. And so I'm going to go ahead then and end our discussion on recombinant DNA. Remember, there is no homework due for this section. I hope you guys have a good rest of your day, and I'll talk to you guys again soon.